Hello students, welcome back to Nucleus Neet Academy and I am Kiran Kumar, your biology NEET faculty. Kya hal chal? How are you? Are you doing fine? Are you preparing for your NEET exam? Great. So today students, we are going to start another important topic from chapter Biomolecules, that is proteins. Have you heard the term protein? Yes, protein is a very important nutrient which is present in our diet, and our body requires proteins mainly to keep ourselves healthy and strong. Okay, do you eat protein rich food? Yes, we all eat protein rich food like eggs, meat, fish, pulses. By eating this protein rich food, we can keep our body healthy. Okay, so why protein is required for our body? In fact, our body itself is made up of lot of protein. So look at the skin. Our skin is completely a protein, and the nails which are present at the tip of our fingers, these nails are also a kind of protein, and the hair which we grow on our head, this hair is also kind of protein. So now imagine if this much of protein is found outside our body, and what will be the role of protein inside our body. So the proteins play several important roles in maintenance of our body functions. So let us discuss about this wonder molecule called protein. So scientifically, what is a protein? What is definition of a protein? Proteins are heteropolymers of amino acids. Proteins are heteropolymers of amino acids. What is a heteropolymer? Heteropolymer means it is a large molecule which is made up of several small units called monomers, and the monomers are nothing but your amino acids. So I will explain you with a ball and string model which I have prepared for you students. Look here, imagine that this plastic ball is a amino acid. Okay, this is one type of amino acid. It is attached to another type of amino acid by a bond. What is this bond called? It is called peptide bond. Okay, now it has become a dipeptide. Second amino acid is going to form a bond with a another type of amino acid, third amino acid by peptide bond. Third amino acid forms a bond with fourth amino acid, fourth with fifth amino acid, fifth with sixth amino acid. So that results in formation of a long chain of amino acids attached to each other by which bond, students? Yes, you are right, peptide bonds. Now we call this chain of amino acids as polypeptide. What is it? Polypeptide. Next, I will ask you a question. Are all these amino acids present in a polypeptide same amino acids? No, they are all of different types of amino acids. So that's why we call the protein as a heteropolymer of amino acids. Understood the concept? Now let us see. Proteins are made up of one or more polypeptides. So our proteins can be made up of one such polypeptide or many this type of polypeptide chains. Proteins are made up of one or more polypeptides of amino acid residues connected by peptide bonds. So the bonds which connect the two amino acids, they are called peptide bonds. You can also write them as CONH bonds. And what does the proteins made up of? Proteins are made up of 5 different types of elements, namely carbon, hydrogen, oxygen, nitrogen and sulfur. These are the basic 5 elements which are present in almost all the proteins. Understood the concept, students? Now look here. The proteins, some are soluble in water. Some kind of proteins can easily dissolve in water, whereas others form colloidal solutions. What is a colloidal solution? So it is a solution where the protein particles, minute protein particles are uniformly dispersed throughout the water. Then we call it as colloidal solution. The best example is your milk. In the milk, there will be tiny protein particles which are uniformly distributed. Those protein particles are nothing but casein protein particles. Understood? So milk is a colloid. Okay, it is a colloidal solution of casein protein. Next, chemical property of protein. Proteins are amphoteric because they show both acidic and basic characters. Now let us see some Interesting examples of proteins. Collagen. Collagen is a structural protein which is present in ligaments and tendons. So students, you might have studied tissues, animal tissues. So in our body there will be a tissue called a ligament and tendon. In these two structures, in these two tissues, ligaments and tendons, support and structural strength is very much required. That's why there is a protein called collagen which provides structural support to these two tissues, ligaments and tendons. And of course, collagen is the second most abundant protein in the living world. Okay, next is keratin protein. So just now we have discussed, it is a structural protein present in our hair and also in our nails and also in the claws. So birds have claws, tigers have claws, right? So in that class also we can find keratin. Keratin is also present in the horns of animals and also in the hooves of animals. So in ungulate animals like horses and buffaloes, there will be hooves. These hooves will protect their toes like this. So these hooves are also made up of protein called as keratin. Okay. Next is another protein, albumin. Albumin is a very important globular protein present in our blood. Albumin protein present in the blood, it maintains osmotic pressure of the blood and also it helps in transportation of different hormones in our body. Insulin is a pancreatic hormone. It controls the blood sugar levels. And the two important proteins, fibroin and sericin. These two proteins are present in the silk. They are called silk proteins. They are responsible for the strength of silk fiber. I will show you here. So this is a nice beautiful silk cloth. Okay. So it is made out of silk threads. And where do we get silk from? Students, silk is obtained from silkworm, right? So the silk fiber is also a protein which contains fibroin and sericin. Okay. Next, amylase. Amylase is a digestive enzyme. It will be secreted by salivary glands and also pancreas. It helps in digestion of carbohydrates. It is also an enzyme. It shows catalytic activity. It is a protein with catalytic activity. It can break the bonds. Okay, this is an enzyme. Next, myosin and actin. These are the proteins which are found in the meat which we eat. So chicken, mutton. So these fleshy food items which we eat contains myosin and actin proteins. Okay, these two proteins are part of muscle fibers which help in contraction of muscles. Okay, next is immunoglobulins. Immunoglobulins are defense proteins. They are present in our blood. They will fight against disease-causing germs in our body. Immunoglobulins can be also called as antibodies. What is another name of immunoglobulins, children? They are also called antibodies. These proteins, they are going to defend our body. They are going to protect our body from disease-causing germs. So like this, there are plenty of proteins which have variety of roles in our body. So now let us discuss what are amino acids. Students, now let us discuss about amino acids in detail. What are amino acids? Amino acids are organic compounds with NH2 amino group and a COOH, that is carboxylic group, attached to same carbon. Okay. So in the amino acid molecule, there will be 4 substituent groups occupying the 4 valency positions. They are hydrogen, carboxylic group, amino group, and a variable group called as R. Okay, there are 20 types of amino acids in the proteins. Now let us see how a amino acid molecule will be formed. Any type of amino acid you take, it has a carbon at the center called as alpha carbon. This is called alpha carbon. The alpha carbon has 4 valencies. It has 4 valencies, right? So in one valency will be occupied by carboxylic group, and one valency will be occupied by your amino group, and one valency will be occupied by variable group R, and the fourth valency will be occupied by hydrogen. So this is a general structure of any amino acid. Okay, since the amino acids have a central carbon called alpha carbon, they can be also called as alpha amino acids. They can be also called as alpha amino acids. Okay, now let us see some properties of amino acid. So in an amino acid, the carboxylic group can easily donate a proton. It can donate off its proton. So thereby a carboxylic group is responsible for acidic character. Okay, and amino group, it can accept a proton. It can accept a proton and change into NH3, which is a basic compound. Hence, a amino group is responsible for basic character of amino acid. Since an amino acid is having both carboxylic and amino group, which are responsible for both acidic and basic characters respectively, amino acids are amphoteric molecules. They are amphoteric. They can be amphoteric molecules. That means they can exist as both acid and base at the same time. Okay, so a variable group can be any type of compound or can be replaced by any type of variable compound. Suppose if in the place of R there is another carboxylic group, then definitely the amino acid is going to get acidic character. If a variable group is occupied by again another amino group, then definitely an amino acid will show basic property. Understood? Okay. Now let us see the 20 different types of amino acids. 20 types of amino acids. 20 types of amino acids. The 20 amino acids which are present in the proteins can be classified into 7 groups based upon their chemical properties. Neutral amino acids, there are 5 in number: glycine, alanine, valine, leucine, isoleucine. These 5 are neutral amino acids. Acidic amino acids are 4 in number: asparagine, aspartic acid, glutamine, glutamic acid. Basic amino acids only 2: arginine and lysine. Sulfur containing amino acids are cysteine and methionine. These 2 amino acids have sulfur in their structure. Next is alcoholic amino acids, 2 in number: serine and threonine. Aromatic amino acids are 3 in number: phenylalanine, tyrosine, tryptophan. And heterocyclic amino acids are 2: histidine and proline. So total 20 amino acids. Let us count once. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19 and 20. These are the 20 amino acids. All the proteins are made up of these 20 amino acids. Come on, copy it down, students. Students, now let us see another interesting property of amino acids. The same amino acid can behave as a cation, zwitterion and anion. And how it is possible, let's see. When an amino acid is having NH3, which is a basic group, then the entire amino acid will behave as a cation. When the same amino acid is having the basic NH3 group and acidic COO minus group, then the same amino acid will behave as your zwitterion. When amino acid has only one acidic group, then it will act as anion. So from this we have learnt that the same amino acid can exist in 3 ionic forms: cation, zwitterion and anion. So that simply depends upon the pH of the solution in which that particular amino acid is present. So amino acids have ionizable nature. They can undergo ionization depending upon the pH of solutions in which they are present. Structure of amino acids can change in solutions of different pH. The structure of amino acids depend upon the pH of solution, and the pH at which an amino acid cannot move either to cathode or anode, that particular pH is called isoelectric point. Okay, so this is about a wonderful property of amino acid. Look here, a cation can form a zwitterion. A zwitterion can again change into anion, and anion reversibly again it can form zwitterion. A zwitterion again can form a cation. This is one of the interesting properties of amino acids. Take down this, students. My dear students, amino acids can be grouped into two types based upon their essentiality. Essential amino acids, non-essential amino acids. What are essential amino acids? Essential amino acids are those amino acids which cannot be synthesized by animals in their body, and they are obtained from plants. Those amino acids which animals cannot produce within themselves, within their body, and they have to take those amino acids from external source in the form of food from the plants, then we call those amino acids as essential amino acids. And there are 10 essential amino acids. What are those 10 essential amino acids? Valine, leucine, isoleucine, phenylalanine, threonine, lysine, tryptophan, methionine, histidine, and arginine. So these are the 10 essential amino acids. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 10 essential amino acids. Non-essential amino acids. These are the amino acids which can be synthesized by animals in their own body and may not be required in diet. So if the essential amino acids are provided to me, then my body can synthesize the rest of the 10 amino acids. Then we call those 10 amino acids as non-essential amino acids. They need not be compulsorily taken from plants. So even if it is obtained, it is well and good, but even these amino acids are not available, then no need to worry. My body is going to prepare them anyway by using essential amino acids. What are those 10 non-essential amino acids? Your serine, aspartic acid, cysteine, glutamic acid, proline, glycine, alanine, asparagine, glutamine, and tyrosine. These are the 10 non-essential amino acids. So they doesn't matter even though we don't have them in our diet. Okay, so let us see some structures of some simple amino acids. The most simple amino acid is glycine, is your glycine. You can see here the structure. There is alpha carbon, there is a variable group H. In the variable group there is a hydrogen, then it is called glycine, the most simple amino acid. It is the most simple amino acid among the 20 amino acids. Now alanine, another amino acid. Look here, the variable group position is occupied by a methyl group, then we call that amino acid as alanine. So here the variable group is occupied by hydroxy methyl group, CH2H group, then this amino acid is serine. So the amino acid name depends upon the variable group. Depending upon the variable group, we identify different amino acids by their structure. So this is about essential, non-essential amino acids, and the 3 amino acids which are from your NCERT textbook. They are going to come in your NEET examination. So you have to memorize the 3 structures of these 3 amino acids: glycine, alanine, and serine. Copy down this, students. Students, now we will discuss about structure of protein. Structure of proteins. Proteins have 4 levels of organization in the structure. Proteins can organized into 4 different levels of their structures. The basic structure and the fundamental structure of protein is the primary structure, and afterwards the primary structure can give rise to secondary can give rise to tertiary, then quaternary structures. So there will be 4 levels in the organization of structure of the protein. First, let us start with the primary structure, the basic structure of protein. What is the primary structure of protein? It is a linear sequence of amino acids forming a backbone of protein. In a primary structure, what you can see several amino acids which are forming a linear sequence, just like this. The first amino acid, it is forming a peptide bond with the second amino acid forms peptide bond with the third with the fourth with fifth with sixth, and so on. Because of this repeated peptide bond formation, a continuous chain of amino acids is formed, what we call it as polypeptide chain. So in a polypeptide chain, you can find different types of amino acids which are linked with each other by peptide bonds. So this is like a backbone of protein. So this is the primary structure of protein. Okay. In a primary structure of protein, towards the left side, the first amino acid will have a free amino group. This first amino acid will be called as N-terminal amino acid. Whereas on the right side, the last amino acid, it will end with a free carboxylic group. This last amino acid will be called as the C-terminal amino acid. Okay, so now once again our ball and string model of polypeptide chain. You can see here, this is the first amino acid. It is linked with the second by a bond called as peptide bond. The second amino acid linked to third amino acid by one more peptide bond. So between the 3 amino acids, how many peptide bonds are there? You are right, only 2 peptide bonds are there, right? So like this, the third with fourth with fifth with sixth. So several amino acids, when they are linked to each other to form a long chain, this is called polypeptide chain. This is what is called the primary structure of protein. Understood? Yes. Now let us see how peptide bonds are formed between adjacent amino acids. Peptide bond formation. Take down this heading. Here you can see this is a amino acid with alpha carbon, amino group, carboxyl group, C double bond O, OH. Here is another amino acid with COOH and NH2, N, H and H. Okay. So when 2 amino acids come close to each other, then a reaction will take place called as condensation reaction. During this condensation reaction, a water molecule will be eliminated. Look here, the OH group from carboxylic carbon, the hydrogen from amino group. So this OH and H will combine with each other and will removed as a water molecule. This is called condensation. So because of condensation, removal of water molecule, a bond is formed between the carboxyl carbon and amino nitrogen. This bond we call it as peptide bond. You can see here, between carboxyl carbon and amino nitrogen, a bond is formed. This bond, what we call it as peptide bond. We can also write peptide bond, represent peptide bond like this, CONH bond. So this is how a peptide bond is formed between 2 amino acids, and several amino acids can be linked by these peptide bonds. Understood, students? Copy down. This is very important concept for your NEET exam. Students, now let us discuss about secondary structure of protein. Just now we have discussed about primary structure of protein, which means what? A linear sequence of amino acids. Okay, so chain of amino acids will form the primary structure of protein. That particular primary structure of protein is going to generate the next level of protein structure called as secondary structure. So the linear sequence of amino acids, that is called polypeptide chain, when it undergoes coiling like this, or it undergoes folding like this, then that generates a three-dimensional structure of protein, which is nothing but called your secondary structure of protein. Okay, the secondary structure of protein is the polypeptide chain coils or folds to form a three-dimensional form. Okay, the secondary structure of proteins were discovered by Linus Pauling. Linus Pauling discovered alpha helix and beta pleated sheet secondary structures of proteins. I repeat, scientist called Linus Pauling. He have discovered alpha helix secondary structure and beta pleated sheet secondary structure. Two different protein secondary structures he have discovered. Now let us see alpha helix structure. In an alpha helix structure, a single polypeptide chain which is made up of different types of amino acids like this, it undergoes spiral coiling like this. It undergoes spiral coiling in the form of a spiral. Okay, it is a right-handed coiled spiral. You can see over here, this is called the alpha helix structure. So because of this spiral coiling, a spring-like structure is formed. We call it as alpha helix protein structure. Here you can see within the same polypeptide chain, between the different amino acids, hydrogen bonds are formed. Hydrogen bonds are formed. So what are these hydrogen bonds are called? These hydrogen bonds are called intra-chain hydrogen bonds. Intra means what? Within. Within the same polypeptide chain, between different amino acids, hydrogen bonds are formed. That's why they are called intra-chain hydrogen bonds. Alpha helix secondary protein structure, it is present in keratin protein and also in myosin protein. Keratin protein is present in our hair, nails, and also present in the horns of animals and also in the hooves of animals. Okay, myosin protein, it is present in the muscle fibers responsible for contraction of muscles. Okay, let us move on to beta pleated sheet. Here in this word itself there is sheet. That means it is in the form of a sheet. The protein secondary structure has sheet-like appearance. Here you can see a polypeptide chain which is made up of different types of amino acids like this. It undergoes folding. Look here. It takes a hairpin bend and moves forward, again in reverse direction. It moves, takes a bend forward, again a bend backward, again a bend forward. So this pattern repeats again and again to form a sheet-like structure which is called beta pleated sheet. Here you can see between the different amino acids of same polypeptide chain, hydrogen bonds are formed. That's why they are called intra-chain hydrogen bonds. So beta pleated sheet is one of the stable structure which provides stability to different types of proteins like sericin protein and fibroin protein of silk fibers. You know who produce silk fiber? Silk fiber is produced by silkworm, right? So the beautiful silk clothes which we wear are obtained from silk fiber, right? They contain sericin and fibroin protein. So now let us move on to triple helix structure of collagen protein. Collagen is the second most abundant protein in the living world. So in animal kingdom, it is the most abundant protein. The most abundant protein in the animal world is collagen. The second most abundant protein in entire living world is again collagen. Who is the first most abundant protein? Can you tell? It is rubisco, ribulose bisphosphate carboxylase oxygenase. It comes in photosynthesis. Okay. Now look here. In this collagen structure, triple helix structure, we can see total 3 polypeptide chains. This is a polypeptide chain. This is another polypeptide chain. This is another polypeptide chain. So these 3 polypeptide chains, they are coiled over each other. They are coiled over each other tightly. So generating a structure called a triple helix structure. Here how this particular structure gets stability because of hydrogen bonds. Look here students. Hydrogen bonds are formed between the amino acids. They are formed between the amino acids. of different polypeptide chains. So this is one polypeptide chain, this is another polypeptide chain. Between the two different polypeptide chains, between the amino acids, the hydrogen bonds are formed. That's why these hydrogen bonds are called interchain hydrogen bonds. What do you mean by inter? Inter means in between. In between the two different chains, between two different amino acids, hydrogen bonds are formed. Okay. Collagen protein is present in tendons, ligaments, and cartilage tissue of animals. Now, the most important point. In these secondary structures of proteins, what type of bonds will be present? Secondary structures are stabilized by intra or intermolecular hydrogen bonds, which are formed between amino acids of same or different chains. So, this is a very important statement. Copy down, students. That's all about secondary structure. Now, we will move on to tertiary structure of protein. Students, now let us see tertiary structure of protein. A tertiary structure, it is a three-dimensional arrangement of protein formed by folding and coiling of secondary structures. The secondary structures undergo folding and coiling repeatedly one over each other to form a complex three-dimensional structure, which we call it as the tertiary structure. So, we can visualize tertiary structure like this. Suppose imagine this is a alpha helix. This alpha helix, it continues as beta pleated sheet. The same polypeptide chain, it continues as a beta pleated sheet. Again, it continues as alpha helix. So like this, the secondary structures, they coil and fold repeatedly in different patterns to form a complex three-dimensional structure, which we call it as the tertiary structure of protein. And this tertiary structure of protein, it is stabilized by 5 different types of bonds. Look here, the tertiary structure is stabilized by hydrogen bonds, both intra and intermolecular hydrogen bonds. Disulfide bonds, these are formed between the sulfur containing amino acids cysteine and methionine, SS bonds, and ionic bonds, hydrophobic interactions, van der Waals bonds. So these are the 5 bonds which are going to stabilize your tertiary structure of protein. All the enzymes which are present in the living organisms have their tertiary structure. All enzymes are tertiary proteins. Next, the biological function of the protein depends upon the tertiary structure. So if a protein is showing any particular biological function, it is because of its a tertiary structure. Suppose if tertiary structure is disturbed because of any reason, then the protein is not able to show its biological function. Understood? So myoglobin is a pigment, oxygen carrying pigment. It is present in the muscles. It is the first protein to be discovered as tertiary structure. Okay. So copy down this, students. Students, now let us understand quaternary structure of protein. The last organization of protein structure, quaternary structure. It is formed by 2 or more polypeptides called as oligomers. When 2 or more polypeptide chains called oligomers, when they are held with each other by bond formation like this, it gives rise to a three-dimensional structure, what we call it as quaternary structure. So best example we can take hemoglobin. So hemoglobin is a blood pigment, or hemoglobin is the first protein to be discovered as quaternary structure. If you analyze the hemoglobin quaternary structure, you can find there are 2 alpha polypeptide chains, 2 alpha polypeptide chains, and there are 2 beta polypeptide chains. Okay, you can see here, this is one alpha peptide monomer, this is another alpha peptide polypeptide monomer. Okay, so this is one beta chain, this is another beta chain monomer. So total 2 alpha monomer chains, 2 beta monomer chains, they all assemble in a particular three-dimensional form to generate the structure of hemoglobin. So because of that particular arrangement, three-dimensional arrangement of two alpha chains and two beta chains, the hemoglobin will get its property of carrying oxygen and carbon dioxide. Okay, so alpha polypeptide chain is having total 141 amino acids. Similarly, this particular chain is beta chain. It is having 146 amino acids. So total 2 alpha 2 beta chains will come to over 574 amino acids. In a hemoglobin protein, there will be total 574 amino acids. Understood, students? And what are the bonds that will stabilize this quaternary structure of proteins? The monomeric units which are present in the quaternary structure are held by non-covalent bonds like hydrogen bonds, hydrophobic bonds, and ionic bonds. So these are the three types of bonds which stabilize quaternary structure. Understood, students? Copy down. Last but not least, we are going to wind up with some protein examples and their functions which are given in our NCERT syllabus. First, collagen protein. Collagen protein shows a triple helix structure. It is an intercellular ground material. It will occupy the space. It will fill the space present between the cells in tissues of tendons, ligaments, and cartilage. Okay. Trypsin. Trypsin is an enzyme which helps in digestion of proteins. Insulin, it is a hormone secreted by pancreas. It is a hormone, a chemical messenger which controls the blood glucose levels. Antibody is a immunoglobulin protein. It will fight the disease causing germs. It will fight the infectious agents present in our body, and antibodies are produced by white blood cells. Receptor protein. Receptor proteins are present on the sensory cells. They help in sensory reception. Mainly you can find these proteins in our ears, on our tongue, okay, in the sensory organs. GLUT4. GLUT4 is another protein. It is a transport protein. It helps in transport of glucose into the cells. Okay, so these are some examples of proteins. So chemically, proteins, they are made up of 20 different types of amino acids, but their functions may differ from each other. Some of the proteins can act as enzymes, some proteins can act as antibodies, some proteins can act as transporters of molecules, and some proteins can act as hormones even. So like this, proteins can have variety of functions. So with this, I am going to conclude the topic proteins. So best of luck, students. Keep preparing for your NEET examination. We will meet in another class with another super interesting topic, that is lipids. So here I stay. Keep preparing. All the best.